Let us start, please. So, on the last presentation, Vitaly explained you all things about spot-based protocols like Spatial Transcriptomics, Visium, like there is also SlideSeq, and also some new emerging things. And as you got, they basically collect multiple cells in the same spot, so you know approximate location, but you lost single cell resolution. So they have very nice properties because they measure all genes in your transcriptome, and then you can, well, basically it's like de novo discovery where you measure all the genes and like single cell RNA sequencing. But like not having cells is a major drawback, and in parallel there was a technology developed which measured individual molecules. So it's not just single cell resolution, but spatial resolution, single molecular resolution. So the idea is that your data looks like this. I don't know how much can you see from there. Like basically here you have like individual dots of different color, and they form this kind of clouds. I don't know. Can you see it from back there? Yeah, cool. So the idea here is that you use imaging technologies to basically first you do some like FISH, fluorescent in situ hybridization, where you take like some fluorescent molecules and attach them. They're specific to certain types of RNA, so you can measure different RNAs and they start like you may see them with some colors. So after that you do microscopy and you see individual molecules based on their color. And your raw data looks like this. So basically you have X position, Y position, and the gene identity. Based on this, you of course can plot single molecules and analyze those. So one thing this data does not provide though, and it will be important later, is cells. So you may see like individual molecules here, you may see clouds, but you don't know where cell boundaries are. So basically you can only guess based on these clouds. So to solve the problem, these kind of protocols usually measure additional stains, like DAPI stains, membrane stains, like full body, like poly-A stains. And here you may see like DAPIs, like each blob is a DAPI. You may use different imaging segmentation techniques to segment those DAPIs and get some kind of segmentation. Like the authors of the paper claim themselves that the quality was suboptimal, and like that's Stan Linnerson lab or some FISH protocol, and you may see like boundaries here do not really match molecules. So that's why people started measuring additional stains like membranes, and that helped to assign molecules to individual cells. The whole workflow of working with this data looks like the following. These protocols, as they require you to design probes for different genes and like put them and measure them, they're really restricted in terms of the number of genes you can measure. I will show you later, but basically it's something from 20 to 10,000s, but 10,000s are super rare, so on average it's like few hundreds, like 100, 500s in that range. And so you usually you would have to get some single cell RNA sequencing data understand what you want to study, like which cell types do you expect, what are their markers, what are the processes you want to study, and what are their markers, and then design a gene panel which would both separate those markers and be feasible in terms of marker design. Because as you all probably know, like with biological experiments, there are things that are simple and things that are not. And some markers just, for some genes we can design probes easily. For the other, they're like, for example, maybe very abundant. And if we have a probe for this individual gene, then it would just highlight all the cells, and you would not see any other probes. It just would be way too bright, so it would basically destroy the whole experiment. After you have the gene panel, you do in situ sequencing or hybridization. Basically, you use those genes to capture cells, to capture molecules, and different protocols do it differently. I will not go into details on that. Then there is spot calling, which basically you have your image data and you have individual blobs which are like spots. But of course, it's not trivial how to, well, distinguish those. So it may be noise, it may be like just multiple spots in the same place. So there is a lot of image parsing going in there. Usually protocol developers deal with it themselves. So basically they design pipelines, they provide you with the software, so you don't need to care about that, but still need to keep in mind that this process of spot calling is the major source of problems. It can, for example, good algorithm can increase yield of a protocol like several folds, and bad algorithm would make your like false positive rate in terms of detecting molecules like 50%. So it's something really crucial, but something you rarely have control for. You also need to keep in mind that like how these protocols work. For example, like we have like 3-4 colors at the same time, but we want to measure 10,000 genes. So how would that be possible? And that can be done using what they call like sequential in situ hybridization, where you, for each molecule is barcoded with a set of colors, like 3 on this picture, but it might be much longer. So for example, you assign on different rounds of your experiment, you assign different colors for different molecules, and you do it kind of randomly, and then you like image first set, wash away the fluorophore, like add another one, wash it again away, and then each gene is encoded as a sequence of colors. So it's also kind of sequencing, and it has surprisingly many similarities with usual cell barcodes, because there can also be like errors, there can be collisions. So if you do not design it properly, one gene, because of an error, can be recognized as another gene. So yeah, quite an important step, but that's something again pre-designed for different protocols. And then you can, each gene has its code, and you understand molecule by its code. But there are also like problems that you do multiple rounds of imaging, and your data does not overlap perfectly. So you may see here, hopefully you may see that your slides kind of shift between rounds of microscopy. So you need to align them and align them on the level of single molecules. So again, very like a place where it's easy to make mistakes. But after that, you finally get your like data for each your molecule, and you have gene code and coordinates for each molecule. Do you have any questions at the moment about this, like how the data looks like, where it comes from, and these kind of sequencing protocols? I will show you later. It's very different between protocols, but like broadly speaking, between 20 and 10,000.
Are the barcodes different for the same gene?
Do you mean can the same gene have different barcodes? In some protocols, yes. Particularly this Tenix Xenium, it has multiple probes for some genes and single probes for other, but that's uncommon. Do you have, yes?
Do the tissues need to be fixed before you do this? Or?
Yeah, totally. Tissue needs to be fixed because you might do a lot of rounds of microscopy on that. And I don't know about all the biological preprocessing there, but I mean, you definitely cannot do it in live tissue.
Are there any technologies that are compatible with protein staining, where you do multiplex imaging to get RNA plus protein?
Yes. I don't know about all of them, and I will bring it later, like at the very end, but technically, yeah, there is a trend that you try to do as many stains as possible, and some people, like Jeff Moffitt, tries to do like hundreds of stains. And there you can measure proteins, you can measure different markers. So Murfish is definitely compatible. Nanostrings, I think as well. And for the rest, I don't know, but they want to be at least. So that's like some emerging trend. Other questions? All right. So 2 protocols. Basically, there are 2 current popular use cases. And I mean, ideally it would be only one, but because we are all restricted with money and protocol complexity and each protocol is not perfect, we can mostly split them in 2 categories. First is cell typing. So basically these are protocols which have small number of genes measured, like between 20 and 100, for example, and it's only enough to get cell type markers properly. Or like some cells process markers if you want to study a particular process. With that you can study like tissue organization, for example, what cell types are where, like cell type co-occurrences, for example, if you expect in, what was that, Alzheimer or something like that, where during the disease, you don't necessarily see expression changes. Well, you see them, but most of all, you see how cell types create different structures and reorganize. So that's basically that you can do with very few markers and just seeing on how cell types are related to each other. Also what Vitaly showed on his last slides. Compositional analysis, again, using spatial technologies is much better than single-cell RNA-seq if you want to count cell type proportions. Because for single-cell RNA-seq, when you do experiment, it really screws your data up. For example, you froze the tissue, and then 80% of T cells are dead. So all the preprocessing really changes cell type proportions based on how cells survive through the sequencing. While here, you can actually see molecules and cells, and they are what they used to be, so they are not distorted as much, and there is no process of subsampling. So like in single-cell RNA-seq, you put your cells together, you put them all through FACS sorting and select only small portion of cells, while here you see the whole slice. So of course it's one single slice, I don't know, maybe 2 millimeters by 2 millimeters or 2 centimeters by 2 centimeters if you are really rich. But that's something, well, you see all cells there. And finally, if you do these cell typing protocols with very few genes, they usually have very high capture rate. Much higher than single-cell RNA sequencing, 40%, 60%, something like that. Sometimes up to 70%, if I remember correctly. So yeah, you may do gene quantifications almost on the level of classical single molecule fish where you do it for a single molecule, but like 95% capture rate. But here you can like select 30 of them. Quick remark on the capture rate. People, for whatever reason, I don't know where they took the numbers, but they say that like Muirfish or some other protocols have like 95% capture rate. Like, that's total bullshit. They don't. I mean, they have something much smaller, and usually the more genes you measure, the smaller your capture rate is. So yeah, be careful when you read the spatial protocols have like close to 100% capture rate. Like, totally untrue. Second type of protocols, it's about de novo analysis, like something we use in single-cell RNA-seq, basically where you measure, for example, 1,000 genes or like at least 500 genes, or if you are like in a Muirfish lab, you can really measure 10,000 genes or 30,000 genes. And that allows you to see a lot of de novo processes. So, well, of course, you may just put a lot of genes and hope to capture cell types without doing much of gene panel selection. You may try to discover new cell types, the ones you did not observe in single cell data. People are trying to do that, though it does not work as well because, you know, having more genes really helps. And here the data is much noisier. You can study cell processes in space, for example, like cell migration. You may really see how they're located and what genes are associated with migration, so you don't need to know them in advance. Subcellular structure, if you have a lot of genes, you can clearly see, I will show it a bit later, how nuclei is different from cytoplasm, and hopefully we would see further like subcellular structures, though people tried, some successes are there, but as protocols emerge, we will see more and more of that. And finally, cell-cell interactions, a very weird topic. I will speak more about that later, but like people are really trying to do that, really want to do that. I also want to have that, but quick spoiler, we are not there yet. So protocols for cell type quantifications, it's like ISS from Sweden, what was his name? Never mind. Like Alice Nassim-Fish from Alien Brain Institute, OSM-Fish from St. Linnerson Lab, Muirfish, some modifications of Muirfish, and StarMap, again, some modifications of StarMap. So MMSE, number of transcripts per gene, is usually on the scale of like 50 to 100. And they also vary a lot with number of like genes per cell, genes measured, well, genes measured also kind of the same. And yeah, number of cells as well. So for example, ISS, Mads Nielsen's lab, it's very interesting idea how to take, I don't know, like just 100 genes, but only like really 5 to 10 molecules per cell. So, you know, like super few, like very sparse cells. You cannot see anything, but they can make a lot of cells. And I think either they were planning to, or like they actually did, just sliced the whole mouse brain and yeah, sequenced it all. So you can do a lot of cells there. Muirfish, like that's one of the best protocols. I will comment on more about that later. Again, they do really a lot of cells and also a lot of genes, but very expensive unless you are like in some custom Muirfish, you know how to develop it, and still you need a lot of money. Protocols for de novo analysis, like most popular, again, some modifications of Muirfish. VisGen, which is a commercial version of Muirfish. Cosmex by Nanostring, and 10xenium, like VisGen, Cosmex, and 10x, they emerged just last year, and well, in the current version, Cosmex provides you 1,000 genes but they are all preselected for you, so you cannot really modify them. So whatever tissue you are, it's the same 1000 genes. This gene and Xenium give you like around 500 genes, but if I remember correctly, you can modify them at least somehow. And yeah, there are also different properties like number of molecules per cell. It's much higher in Cosmics data. Like here, it's even dataset where the difference is not so transparent, but like we have data with thousands of molecules per cell. And yeah, number of genes per cell also varies, but it's not as important because, I mean, it does not play big roles in Cosmics having a lot of genes because it's super noisy, a lot of false positives. So having a lot of genes per cell, it usually means that you just have a lot of false positive genes per cell. And effective number of genes in the protocol, like in our estimate, is around like still 500s. So the number of 1000 is not exactly applicable. So yeah, answering about my favorite protocols, we did a lot of benchmarking on these three. We are still in the process of that, but Xenium seems to be like the one with the clearest data, and you can see good separation of cell types, low false positive rate, and yeah, but also pretty expensive. Cosmex, like a lot of genes potentially, they do membrane stains, like some protein stains, but false positive rate in our tests was like 50%, close to that. So it's so noisy that it's hard to make sense of that. And this gene is both noisy and few genes. So yeah, like in the current state, I will record it maybe, but this gene was not impressive. Let's say that. Any questions on the protocols? Yeah?
Maybe a clarification on previous point. Are you saying the current best is probably Xenium?
I'm saying that of these three commercial technologies, I would bet on Xenium, though there are pros and cons between it and Cosmex, and we don't have a definite decision yet. But Xenium seems to be the most promising. But if you collaborate with some lab who actually managed to establish Murphy's, like Jeff Moffitt, Shao Wei, there was some other folks from Shao Wei lab, like not commercial with gene, but actual Murphy's, that's usually the best option. But that, I mean, if you want to establish it for your lab and you're not from Shao Wei's lab, it would take like more than a year of just protocol adjustments.
What kind of false positive rates are we talking about in Murphy's opinion?
That really depends on the tissue. I don't have data from Murphy's, so we compared it on lung cancer data because all three protocols independently published lung cancer dataset. We don't have it for Murphy's, so I cannot tell you exactly. I know that in most protocols, it really depends on the tissue and your probe selection. Well, except for Cosmex, because the probe set is predefined, so there it's always high false positive rate. But on Murphy's, I worked with the data where it was really close to 0. I heard of the data where it was close to 10, maybe 20%, but yeah, it depends on the tissue.
So it varies, I guess also depends on execution.
Yes. Yeah, that is why you would need to spend 1 year to set it up in your lab, because, I mean, we collaborate a lot with Jeff Moffitt's lab, and like while we were publishing a paper, I first got the data, and I mean, just in 3 months, it got 3 times better, just by some protocol, relatively minor protocol adjustments. So yeah, a lot of stuff to work on there. Any other questions on protocols? Anyone who wants to use protocols in their lab and don't know what to do? Yeah? Do you have questions on that?
Well, not now, yeah. Like, you said that it's 100 of genes, maybe 500 for Xenium. But how, like, what these genes are? We have, for human genome, we have 20,000 of genes. And they are all pretty important.
Yeah, that's the thing. I showed some time here. So first step basically is select a gene panel. So
And they're all customized.
I think I might be mistaken here. I didn't look in the new commercial model, but I think they provide you a bunch of panels. So you select from, they pre-selected genes for you, like for neurons, for something else, and then you pick out of them. But I might be mistaken here, like that's something I just saw briefly. So yeah, like I think that's what they all struggle with, because pre-designing a lot of probes is laborious and expensive, and they want to sell as few probes as possible, but also cover as much as possible. So I believe it will change with time how they develop protocols. Yep. Sorry. No, go ahead.
How much it is expensive?
I cannot tell you, sorry. I mean, you need a really good microscope, so at least count that, and then also probes, but I don't know the numbers. I know that I know 70% of the lab, I would not expect them to be able to afford that. Vitaly?
How much should you be concerned with the crosstalk between genes? If you wanted to measure gene X, but your probes bind to gene Y.
Yeah, well, I mean, non-specific hybridization, to my understanding, is one of the major reasons of this false positive rate. So again, I mean, this false positive rate is a very good number of all kinds of problems, including non-specific hybridization. And as I said, like nanostrings, super concerned, like we're developing methods just to remove some noise because it's terrible. But I mean, otherwise the data is pretty good. And Xenium, like almost no problems, Murphy's almost no problems in many cases. So yeah, I mean, you are concerned with that while probe design, but on designed probes, kind of depends on the protocol. Does it answer your question?
And the question is, how do you remember, let's say, if you are buying stuff from 10x, how do you know that they designed the probes well? You don't really design probes, right?
Yeah, but well...
or if you design probes yourself, how do you know that you did that well?
False positive rate, again, I mean, you take your data, you plot your like do single cell analysis, like classical this plot, and then you plot markers, like marker of endothelial cells. Would it be only in the endothelial cells, or would you see it everywhere? And on Cosmex, you would see it everywhere, but here is the highest concentration, so there is like no cluster which do not express marker of endothelial cells. Then you know is that you messed up something.
And my question is, how do you choose probes before you do those experiments?
Oh no, you don't. I mean, like, I cannot answer it precisely, but a lot of trial and errors, like a lot. That's an expensive and hard process. So I guess we are ready to go next. So I told you that cell segmentation is one of more crucial problems here. And the problem is with cell segmentation that it affects all the analysis. So like just to summarize, like single-cell RNA-seq analysis, tissue organization, spatial processes, cell-cell interactions, subcellular structure, whatever you try to study, cell segmentation would fuck you up. Like, if you do single-cell RNA sequencing analysis, you have these bridges, and for those who are familiar with RNA-seq, it really is a strong sign of having a doublet. But here, like, the problem with doublets is different. We are almost out of paper. Okay. Where do you have the markers? Thank you. Yeah. Right, cool. So basically, imagine that you have two cell types. So each dot is a molecule here. So that's one cell from one cell type, and that's another cell from another cell type. And for for whatever reason, you put a boundary like that. So it's not exactly a doublet, but this cell would technically occur between the green cell type and the red cell type. So, I mean, let's say that it will be something here, for example. So, yeah, a lot of doublets, and sometimes you don't know what are doublets, what are real cells, so quite a problem.
I have a question.
Yep.
In that case, isn't it possible to somehow correct that by repositioning the segmentation?
Well, that's the whole segmentation problem, right? Like, depending on how you make the segmentation. So your goal is to make a segmentation which does not make such mistakes. But they're not always easy to recognize, because sometimes you have a slice, and in this slice you have one cell, like here, forget about the green part, like here, but from the third dimension, or because of whatever weird cells sitting close to each other, you have a bunch of molecules here. That happens also a lot, and ideally, of course, you want to say that you kind of have a cell within a cell, and that's a whole different thing, but how would you do that? Depends on the algorithm, and yeah, quite a problem. The second problem is, like, if you want to study tissue organization and special processes. It's a bit harder to grasp, but what you want to know is what cell types do have spatial patterns and what cell types don't. So, for example, here is like a mouse brain colored by kind of cell types, and you may see like here is one layer, another layer, third layer, and as with all things, like, I mean, in brain generally, excitatory neurons have spatial organizations and inhibitory neurons don't. So, well, not as much at least. So here you may see clear layers and also a bunch of inhibitory neurons just all around. But now imagine that a lot of inhibitory neurons also have their boundaries messed up. That would mean that inhibitory neurons in layer 6 have a bit of layer 6 expression, and inhibitory neurons in layer 1 have a bit of layer 1 expression. So basically you may say that you can see clear clusters of inhibitory neurons in layer 6 and inhibitory neurons in layer 1, and they are different. And you may assume that they have spatial structure, but they don't, because it's all a problem of segmentation. Similar thing with cell-cell interactions. Like, people currently publish a lot of papers on cell-cell interactions where they show, for example, exactly that you may see how inhibitory neurons in cell type 6 correlate with layer 6 excitatory neurons. And guess what? They never correct for segmentation errors, and obviously you would see correlation if, if your boundary is set up in a way that it captures both cell types. So that's one of the main reasons we cannot currently reliably infer cell-cell interactions from spatial data, because if we would have a clear segmentation without any artifacts, or at least like 80% without artifacts, then we can say that, yeah, let's just try to estimate correlation between adjacent cells and see how they communicate to each other. But yeah, we cannot do that at the moment. And finally, there is also this new fancy direction of subcellular structure analysis. The main idea is that you take your cell, and then you want to find, for example, we cannot do that right now, but like mitochondria. What if we would measure mitochondrial genes, and we would expect a mitochondria to be somewhere here with a very specific set of genes? So what people do is they basically take these kind of local spots, cluster them. We will do it a bit in a workshop, and see how like spot here is different from a spot here. And if we see some kind of consistent spots, like with different expression, then we would say it's a subcellular structure. But if we have wrong boundaries set, for example, here you may see this yellow molecules got a bit into a blue cell, and so if these cells are often sitting close to each other, you would see consistent patterns of yellow molecules in a blue cell, and you would think it's a subcellular structure, but it's just a part of an adjacent cell. So again, follows positive biological results. So, main reason, like main conclusion from this slide, please be careful. If you ever work with spatial data, if you ever read a paper in spatial data, think about segmentation, like, did people correct for that? Do you correct for that? And most likely the answer is no, and the results are totally not trustworthy. So that's like a major pain.
I have a question.
Yes?
So let's say that I'm reading a paper on spatial transcriptomics, and I would like to understand whether they really did a good job. So what I should look for in that paper?
So usually they perform some analysis, and like a big red flag is that if the authors assumed that the segmentation given by the protocol authors or by them measuring the data is perfect. So like just, people not speaking about segmentation, like not at all, just we took the data from Murphy's protocol and it's, then they didn't do anything, and then you should not trust unless, ideally, of course, you should take the data, load it, see what caused the results, and try to understand if it's a segmentation problem or not. I spoke with some authors of such papers. They just, they don't look at the data. So just you can take the data, take the results, and look at what is the sources of things. Because these kind of problems, it's trivial to spot, Like if you see subcellular structure, just plot a cell, plot a structure, and you would obviously see that it comes from a separate cell. But not everyone does that. So, did I answer?
I have a question.
Yes?
I'm not sure if I understand, because on this picture, there are red boundaries and black boundaries.
Sorry, I was too lazy to make a new perfect picture, but technically, red boundaries here, they show you DAPI segmentation, and black boundaries, they show you adjusted segmentation. So you may see that DAPI segmentation has more problems because it does not account for gene composition, and black has fewer problems, but still some. But that was not the message from the image, but that's what the lines mean. Does it answer your question, or do you have something else?
Yeah, it's okay.
Yes.
If the next section of the cover, how do you rectify this issues with segmentation? How do you deal with it?
How do you do segmentation, or how do you deal?
How do you deal with issues that arise due to bad segmentation?
The problem is that the area is very much under development. So there are no good practices. I will show you some kind of good practice, or like something which is called good practice, but they're not. So here, in case of, it really depends on the problem. Like if you do subcellular structures, then it's the easiest, I guess, because you want to, different kinds of structures. So basically, an output of an algorithm for subcellular structure analysis is a set of structures. And then you can check each of those and say that these are segmentation related and these are not, because, for example, they are very deep within a cell and they make some biological sense. So there you can just filter bad results. With cell-cell interactions, as I said, it's just impossible to do now. So even though there are like, I don't know, few dozen papers, I believe at the moment, who claim to do that, I'm not sure any of them would work actually because of this segmentation problem. Tissue organization is a bit harder because, I mean, like some tissue organization you may still study, like you know that excitatory neurons have layered structure, and you know some cells which migrate, so you can see like broad processes. It's just not all the fine-grained things you may see, and theoretically for that you can also try to remove all doublets. So if you only have kind of pure cells, it would help with such, well, with all analysis, but just those pure cells, you would need to remove, I don't know, 50% of your data at least, maybe 60, 70. So kind of not so representative.
One more question, what do you mean by some interactions?
I will explain it a bit later, okay? Any other questions? So I guess we'll go through a couple more slides and then take a break. So why stains are not enough? Well, I mean, I showed you that we have some membranes and people usually do membrane stains, and here is why they are not enough. Basically, Murphy's data, you may see some cells have very clear membranes here around them, and some cells you can segment perfectly by membrane data. But the others, it's just a white spot, so no membranes were detected. And the problem that it's not just a random signal, but membranes are captured better in some cell types and worse in other cell types. So you may just lose just whole several cell types. For example, in the original OSM fish segmentation, I think they lost like 90% of astrocytes because DAPI was not detected for them. So yeah, just even if you have like perfect membrane segmentation, I mean not perfect, but like good membrane stains, you would get such problems. But on top of that, sometimes you don't have stains at all because you run experiments, maybe imaging part for molecules worked and stainings did not. That happens all the time, so not always you even have stains. On top of that, it's not just membrane you have, but you also need to run some fancy AI methods to get cell boundaries from these. And these methods are still not perfect. I would expect them to be much better, to become much better in the next year or so, because AI is everywhere. It became so good just last couple of years. So hopefully the imaging segmentation problem will disappear. But finally, there is also a problem of just weird biology which we cannot explain. For example, here you may see we have a DAPI and we don't have molecules around it. So like each dot is the molecule, blue signal is DAPI. And here we have molecules, but we don't have any DAPI. Again, here is like kinda super small spot DAPI-like, but yeah, so they don't always match to each other, and that happens all the time. I mean, I was not even looking for this picture, I just took a random picture, and guess what? It is there. So, yeah, well, the claim is that segmenting using stains is not enough. And I will show you briefly a method which we developed, and I mean, I understand that it sounds biased because, well, it's our method, but it's kind of the only real method in the area because we started working on that like back in 2017, and like, yeah, all methods which emerged recently, they kind of try to do similar stuff, but with much more data required for them. So I'll show how we do with that, like what the method does, what is our current take on how we generally can deal with spatial data, on what is there like in terms of other published methods on spatial data, And that will be our next half of the lecture. So if you have any questions, just ask. If you don't, feel free to take a break. So what we can do here, like neighborhood analysis. That's the thing which Vitaly showed you. Like you can take, after your segmentation, after your cell type assignment, you can check basically which cell types often sit together. And this matrix shows you like correlation of those cell types. And there is also a distance plot. So for each like cell type, or here I believe it's like spatial domain, but I mean you can do spatial domains, you can do cell types. So for example, you can take cortex 1 and estimate distance to other spatial domains. So this like plot gives you understanding of how different domains are related to each other. And yeah, simple but helpful. As I said, there are also a bunch of methods on both spot-based and single-cell-based, which try to cluster cells or spots and provide you some domains which are continuous. So that's very close to what I showed you here, but like this method works on the level of single molecules and does not require segmentation, but also, I mean, it's noisier. And if you have segmentation, your data becomes like, your task becomes simpler, so you can like cluster results in space and yeah, get these nice domains. Spatially variable genes, again, they show it here only for spot-based, but there are tons of methods for all kinds. Basically, the idea is that find genes which have some spatial patterns. Because sometimes you may assume that there are, of course, some housekeeping genes or whatever, which are distributed equally across all cells, but you may look for some spatial patterns. Either you may look for genes which, like here, give you like a whole domain, or you may look for genes which maybe like form different spots, or like some like diverse structures. And these are sometimes different algorithms, sometimes the same algorithm can do the same, but the point is like when you have space, you can try to find spatial patterns in genes, and that will give you at least pretty pictures and hopefully some understanding of the biology behind.
Viktor?
Yeah?
Just one comment on collocation type analysis.
Yeah.
One of the issues is how people often do it by just computing correlations of, in case of single cell data, you have different metrics, but if you just compute correlation in abundance of cell types, in certain boxes, locations like what I showed, the problem is that because in majority of the tissue, those two cell types will be absent, the correlations for most players are tiny, and they stop being informative. And this is why I described negative matrix factorization as a way to get that tissue zonality. So it could be that there are pairs of cell types that correlate, but you have to account for the fact that they only correlate when both of them are present. So you have to deal with that aspect. It's not so much to you, but to everyone else, that you have to be careful with not using just variance or just correlation in this context, because there are so many distinct microenvironments.
Yeah, thank you for the comment. It is important indeed. So, yep, thank you. So, the other task they show is spatial deconvolution, which is only relevant for spot-based, so I will not cover it here. And the final task is imputation. Tangram, I guess they again show it for spatial. But yeah, the idea is that if you measure only like 500 genes, you might be really interested in other genes which you did not measure. And what people try to do is imputation. Basically, based on single-cell RNA-seq data, you learn correlational structure of genes, and then you predict some genes based on the data you measured in space. So that's what people propose to do, but I would really warn you against doing that, because the problem with imputation is that it trains some kind of black box model and then predicts, and then you plot it and try to make conclusions about that. But just a simpler and more transparent way to do exactly the same is to take your single-cell RNA-seq data, take your spatial data, align them together, see clusters, and like get back your, well, show how your single-cell data, how it expresses genes similar to what your spatial data expresses. And that would mean that you see some real stuff. So if you show like genes in spatial data, genes in RNA-seq data mapped to spatial data, then that's exactly that. But if you show imputation genes, then it's something weird, because it's just predicted of some models which may not be correct, which you may not evaluate by intermediate results, and yeah, very problematic. Finally, the topic which they did not cover here is cell-cell interactions. I will show you how it's theoretically supposed to work, but again, like, be careful. So imagine that we have multiple cells, and in this case I will be painting each dot is a cell, not a molecule. So like one cell type, maybe it's like cancer cells, maybe it's like neurons who communicate with each other, and the other cell type. So what we want to learn from this is basically you have two pairs of cells here which are close to each other, and we would assume that if it's close, they might be signaling something to each other, and we expect that if cell A signals to cell B, then at least cell B, or maybe they both, would change their expression patterns somehow. So what we can do here is take these blue cells from here and, well, A, B, and the rest, and try to compare expression patterns of A, B versus the rest cells of these cell types. Of course, you would have more than two cells, but just trying this kind of differential expression will show you that they have some distinct patterns, and we can expect that they are like stimulated by this spread cell type. We can also try to, well, we would try to do exactly the same the other way around, just taking these red cells and comparing against other red cells. If we also know that there are some changes, we may try to use some gene databases to understand how they basically, what these changed genes mean, and whether it's like a signal itself or whether it's a consequence of a signal. But that's, yeah, like a next step. So this is a basic like intuition behind ideas, and people try to use, I don't know, 20 different methods just to find these adjacent cells, find their correlation, find like this differential expression between these and the rest, because there are like, of course, the questions like, do this is close enough? What about other cell types? What if there are three cells? So like, a lot of questions around it. But the idea is to find how cells which get signaling are different from cells which don't. And ideally, if we know that, we would know, like, I mean, if we know the signal and results of the signal, we would know, for example, how different drugs can be designed to affect our cells, or maybe how some immune cells can kill cancer effectively while others don't. So we would know what processes we want to induce in our cells to get the results we want. Any questions about this? Now I want to ask some questions from you, but like, do you understand why we need this at all?
To improve the accuracy of this method.
No, I mean like why do we need all these methods? Like, why it is so cool to have spatial dimensions and like estimate these neighbors, estimate these domains?
To improve resolution.
And why is spatialomics? Like, why to use?
I mean, why would you want to use spatialomics at all?
Cell interactions.
Yeah, to
To provide the true image of the tissue.
To provide what?
Provide the most real-life snapshot of transcriptomics of the tissue.
Also that, yeah, okay. Well, I mean, no, thank you. I mean, I'm just asking you because I feel like there is some gap and I feel like I lost some people, so I want just to get connected with you.
Is there a way that you can, if you figure out the number of transcripts in a cell, can you like backtrack into like a single cell RNA-seq?
Sorry, if you have what?
If you generate a count matrix,
Yeah.
with the transcripts that you detect in a spatial omics,
Yeah.
can you like apply the typical scRNA-seq workflow?
Yes, you can, like mostly. So if you would just take spatial data and apply single-cell RNA-seq workflow, you would get like normal results. Where was it? Yeah, like this embedding, like cell type annotation. You can totally get that. In practice, though, Jin Fan, who does amazing job on spatial methods, recently showed that, for example, typical normalizations, like total count normalization, actually distorts your results. I mean, it has some problems. For example, if you have only 2 genes and you divide by sum, then you cannot understand, like it's just fraction of 2 genes. So like the more genes you have, the better this total count normalization works. So in case of 20 genes or like 50 genes or even 500 genes, she showed that total count normalization distorts your data and does not give the best results, but they did not find what does yet. So some of the methods would introduce some artifacts, but generally, like on average, I would say you can use normal single cell methods, and it's not like you have many alternatives at the moment. Any other questions? Yeah.
So probably this in your paper, but I still don't really understand why segmentation is not working with that piece for security result. If you use something like CellTools and it's not competitive enough with the method?
Yes, it's not. I mean, so we did a lot of benchmarking, and so there are cases like here, for example. This is your DAPI signal, and you may see that it's a bit lighter between cells, but CellPulse does not recognize that, and your DAPI segmentation gives you this result, like the purple one, and yeah, Bayesian segmentation gives you much more proper results.
Right, like proper settings, like there's tons of cell masks that are just membrane specific, and then try to, because that I guess it's not working perfectly, but if you would get confocal image of cell margins, wouldn't that be more helpful than that?
like poly A, poly T markers, but that is not really helpful because you just see, I mean, they're too close to each other and finding boundaries there is a hell. You may take something like cell type markers, but then you would need to do a lot of stains and for each marker do segmentation separately. That could work and that would help a lot, but we just don't do enough stains at the moment. And for membranes, I showed you some results, that basically you just don't always detect those. And here is an example of a good membrane. Like if you look at membrane data, much more often it's really messy. Does it answer your question?
Yeah. Other question. Like if you, maybe it's a stupid question, but I imagine there are like these spots with barcodes, and if you were to get like the extreme resolution of confocal imaging and then wouldn't it be easier than to track back the cells that you are getting, or am I just confused?
I'm not sure what you mean here.
Like before, like you have your tissue section, and if you were to digitize it at very high resolution while overlaying on the barcodes to kind of get the idea where they are, wouldn't it be easier than to segment the cells?
It seems like people are unable to do that. I don't know. I mean, just no one does that, so I assume it has some biological problems. A question? Okay. Yeah?
Just, I don't understand quite well, like, what is the difference between the refined domains and the normal domains? Like, you show us in the best practices document that you have two images, one with the refined domains and one with the normal domains.
28.4.
Yeah, 28.4.
24 and 4. I would assume it's... Okay, well, first of all, I don't know exactly, but I feel like it's something very specific to spot-based methods. So usually you have problems with your domains because each spot here has some, has multiple cells, so they are different, and it's not always you can infer domains properly based on spots purely, so you want to assume that each spot may have different domains, so people do some refinement. I don't know about the picture here, but that's my main assumption. Yeah?
My question, because you asked us basically if we understand what the whole thing is good for. So basically, if I see it right, so it's basically that we know exactly, or as good as possible, where sort of the cell types are located.
Yeah.
But this is then based in these protocols on really a small amount of genes in those cells. So if I think about single cell data, there's normally something like, I don't know, gene regulatory networks, I don't know. So some kind of post analysis in the end you can do. Can you do this, or are there some kind of such analysis on this kind of data, or is it finished when we know where the cell types are located?
That's back to the question about protocols, like here. So if you have your protocols with very few genes, like with 20 to 100 maybe, then it's really unusual that you would be able to do some differential expression or gene ontology analysis, unless you measure some genes designed, for example, specifically for one cell type. Maybe like there is a cell type changing from an injury, and then you measure a lot of genes, but from the same cell types. You would lose a lot of cells, but you would capture precise expression of a few genes of one cell type. And then you can do differential expression, like depending on the distance from injury, for example. But otherwise, you would not be able to. In the second case, though, if you have 500 genes, it's it already may be enough. I mean, definitely you would not be able to capture all functional processes, but people do geoanalysis and D-analysis, and it works. Great question, by the way. Any other questions? All right. So now is the last part on what we can actually do with all these methods, which people don't talk much about. Like some people know it and do it, other people don't, and almost no one does it. So the idea is like, I mean, you saw all these fancy pictures. Well, I don't show them. But like the question, like how we can understand what's in our data, at least by eye. And the idea is like, let's take our molecules. Again, each dot is a molecule. Let's, for each molecule, take its neighbors, like 30 neighbors, 15 neighbors, something which is usually on the scale of cell size or somewhat smaller. Then we may assume that it's a pseudo cell, like a tiny cell, you know, we can estimate number of molecules of each gene here and build your usual gene vector, like in count matrix for single-cell RNA-seq. And surprisingly, you can take all those vectors and cluster. So here each dot is this pseudo cell, and actually we have as many dots as molecules in the data set. So it's something really huge in terms of number of pseudo cells. But we can take a subsample if you want. And then we can do clustering, we can show markers, we can do cell type annotation. So technically, we can see what's in our data before any segmentation, before like almost no other analysis. But we can find markers and we can manually annotate it, and we can also map it back and color again by cell type, if you want. So that is the first idea, what we can do like on the segmentation-free level. But what if we take this gene vector, like, I don't know, 20 genes, 500 genes, whatever, and use some dimensionality reduction and 3D embedding. So basically, we reduce the dimensions to like 3D, and as soon as we have 3D dimension, we can convert it to colors, like RGB, red, green, blue, like 3 numbers. So we can convert 3 numbers to a color. And that would give us these pictures where expression of each neighborhood of each molecule is shown by color. So if some molecules like here have the same neighborhood and here have the same neighborhood, they're shown in very similar colors. But if the neighborhoods are very different, you see different colors. So that gives you understanding of your tissue without any segmentations, without anything almost. And you may see it by eye. And that's also what was used to, I mean, I used it to show you previously, like these cells overlap and segmentation problems. So this is this kind of coloring. Using that, you can show pretty pictures, and actually just varying the K, you may set K like something really big, like, I don't know, 500 molecules, like 1,000 molecules, and then it would focus on tissue structure, like here. So you mostly see like just broad layers. Or you may take it small. Oh, yeah, you can also use this to draw pretty UMaps. Really not useful, but pretty. You can take it on the level of single cell, and that would allow you to inspect segmentation boundaries and compare them and understand how good did you do. But also, if you take something like smaller than a cell, then you can see subcellular structure. For example, here is like Murphy's gut data, and you may see like here is a cell, and like it's actually an outer wall of well, stomach, and you may see here like green is nuclei, and then we see polarization towards the outer boundary, and it's kind of gradient, and yeah, it's like cytoplasm, and there are also some, it seems like there are some also further patterns than just these 2 compartments, so like it's a gradient. And that's one thing we can do with these neighborhood competition vectors. how we call them, is basically you can inspect it visually, or you can take, for example, this whole outer wall as a cell type and cluster NCVs within this outer wall. You will try doing that in workshop and see how, like, what kind of inner patterns are there in our cells. On top of that, we can try to interpret what's in our NCVs, like just how are they encoded. So we may take like our data, another data set, and just color it by the first dimension. So NCVs have like 3 dimensions, but we may take 5 or however many we want to. And if we plot it by dimension number 1, in this example we may see that it shows you like nuclei versus cytoplasm just perfectly. So just dimensions of these NCVs show you some biological signal. So dimension 2, for example, corresponds to certain cell types. So here is like this brownish thing. And yeah, you can do a lot of that, but so far the most useful was really like to understand, to find nuclei compartment without any information. Like you don't need even DAPI for that. Finally, as I mentioned before, like it's currently people are working on doing multiple stains. So for example, like it's non-shrink data, they always do PANSUK, CD45, CD3 stains, or at least in all their published datasets. So, but these are images, so you would need to overlay images with your molecule data, hard to visualize, hard to understand. But what if for each molecule we take brightness of each stain in this molecule? You know, it would give us exactly, well, in this case, 3-dimensional vector, so dimension 1, dimension 2, dimension 3. Or if you have more stains, we can take multi-dimensional vector and embed it in 3 dimensions. And that would give us the same coloring, well, same kind of coloring, but you may see in molecules like what stains are there, and if there are correlating stains, it would also be seen. So like blue here signal is like, it seems like it's capturing whatever, some of those. Anyway, so you may map like individual cell, like bright spots, to individual spots on stains. And the more stains we would get, the more useful this technique would be. And we also can like concatenate this staining vector with gene expression vectors and get a matrix which captures both signals in, well, in one data set, in one matrix. No. So, yep, that's kind of it on segmentation-free analysis. And it says it's also time for a longer break. So do you have any questions on segmentation-free or anything which was in the lecture?
I can find that how this embedding works. How do you embed the gene expression again into 3 dimension?
So first, we take molecules, for each molecule its neighborhood, and we call it a pseudocell. Is this step clear?
Yes.
Yep, so we have a gene vector of like how many genes we measured, it's the length of our vector, like our normal count matrix. Then you do like your standard ScanPy stuff like PCA plus UMAP, but instead of 2-dimensional UMAP, which you usually do in ScanPy, you do 3-dimensional UMAP. So is that part again clear?
Yes, so it relies on 3-dimensional UMAP.
Yes, but as soon as you have 3-dimensional UMAP, you may take renormalize each dimension from 0 to 1 and get like 3-dimensional vector each from 0 to 1, and in one function you can convert it to an RGB color, because like if the first dimension is red, the second dimension is green, the third dimension is blue. But RGB colors have their problems because it's called perceptual continuity. The idea is that you want colors which are close to each other be So if you have numbers close to each other, you want the colors visually be similar to each other. And RGB does not have this property, but there are other color palettes which do, and lab space, just the most popular in like this data visualization community. It works a bit weirdly. I don't want to explain that. I mean, like, no reason to do that, but you may consider it like a fancy RGB.
Yeah, makes sense. Thanks for explaining. Sounds like you can use in principle any 3D embedding to visualize for this.
Yes, Yes, we did it as well. Like also, Jeff Moffitt did it for tissue organization. So basically, what he did, like, so imagine you have a segmented tissue and some of your cell types, like maybe here. But you also have cells. So for each cell, you take neighbors, and then you do exactly this idea of NCVs. You embed it in 3 dimensions, and yeah, but you embed proportions of neighbors in each vicinity. And that gives you like spatial domains or like spatial correlations in some way just visually on your data. So you can embed a lot of things like that, and I think that's really underrated idea.
Sounds like a topic for a fun project. Make your own wallpaper.
How about we discuss that and maybe make it for a hackathon? Any other questions? Like segmentation-free things or the whole spatial stuff? All right. So I guess then, thank you everyone, and you know where to find me. And after the break, we will have a workshop where you will take data and manually play with that, see what's inside, see how you can analyze it, and yeah, do some coding.